Hi, welcome to Hard-Boiled Synthesis lecture. I really don't remember at this point. 8? 9? 8. I had to look at my notes. 8. I'm continuing the extraction process. And so far it's been difficult, disheartening. My sadness and my frustration at this point is pretty funny, manifested itself into like a physical challenge. I'm slowly growing a sty. It's been ballooning all week, but today it just really was swollen up. It's just nothing but the best these days. And so, but I'm going to maintain a positive attitude, positive energy. It's Friday. Although at some point I might be like a little bit like Le Chiffre from James Bond, where I might like have a slow teardrop of blood coming from my eye. What's happening is internally I'm steaming from the frustration of extracting these data. That's the take-home message. It's going to be tough. It's going to be tough to pull the numbers you want from studies. But don't get discouraged. Yeah, tackle it as a learning activity where you just kind of learn, oh man, there are way better ways to report your study findings. Again, though, I want to step back. My goal is to not disparage these studies. I mean, these studies are doing what they are. They're presenting things. They're not presenting things so that a meta-analyst will be extracting stuff later on, right? They're just trying to get their results across. But so far, it's been, I had to drop 2 studies flat out because they didn't report things in a way that was useful. Hopefully today it's going to be better. I'm going to extract more data. So a little bit on my progress. I've processed 5 studies out of the 27. Hopefully do 2 more today. I don't want to spend too much time on this today. And let's get started. I mean, what else is there to do than to start pulling out numbers? This is the exciting part of my day. Okay. So let's start off with study number 75, the effects of plant essential oils on escape response and mortality rate. Okay, so escape response is what we want. That's type of way to measure repellency. Mortality stuff, not interested in the toxicity of whatever they're looking at. And let me just double verify that there's catnip in the study. Essential oils from citronella, hairy basil, catnip, and I don't know what that is. So I'm just going to laser focus on the catnip, pull out those outcomes, and move on. Start off with the easy stuff. Start coding the species. So this is study 75. We got 2 species, Aedes and an Anopheles species. So I like kind of seeing this diversity of taxa exposed to repellent activities. Not all mosquitoes are alike in the way they respond to repellents. So in my books, this is always good. And in the end, when it comes to the meta-analysis, if there is a super strong effect of catnip and it's consistent across many species of mosquitoes, then yeah, maybe we are approaching towards the claim that catnip is better than DEET. Although, up to date, right now, with the 5 studies processed, I have not seen that. So we got Aedes, and then we got Anopheles, let's figure out the ages of the mosquitoes tested. originated from Thailand, Bangkok. Larvae were fed with fish food. Adults were kept in screened cages, provided 10% sugar. 3 5-day-old unfed females were used in this. 3 to 5 day old. Okay, that was a confusing sentence. I thought they were like, we used 3 mosquitoes to do this experiment. So 3 to 5 day old unfed females. Thank you. 3 to 5 day old. I'm assuming it's the same for both species. Unfed, so starved. To date, there has been no variation on the feeding activities of mosquitoes, and that's kind of like a standardized thing to do before the experiment. You want the mosquitoes to be responsive, and so you just starve them a bit beforehand. All right, let's pull some info about the catnip. Describe the species, always good. Catnip oil, purchased. I like it when it's that straightforward. What's happening here? All right, concentration tested. Repellents DEET and picaridin, which is another chemical used as a repellent. Were used as standard repellents for comparisons. DEET, 97% purity. Okay, so these are in super high concentrations here. I imagine at some point they must dilute that stuff. I'll just highlight this for now because it means I'm probably gonna have many rows. There's 2 controls, no negative controls, so 2 positive controls. Essential oils were tested against Aegypti using concentration ranges between 5 to 3%, diluted in a solvent. Final test concentrations were used to knock down mortality rates. Nylon netting was cut. Volume test solution was applied evenly to net strips. Okay, and they do have a negative control, which is just ethanol. Whew, confusing. Okay, so they just tested this stuff on nets, I guess. Or they applied the... Okay, they impregnated and infused nets with the essential oils. But there's got to be some sort of attractant. Contact. Okay, so we got two ways in which they measured repellency effects. They have contact, and then they have spatial repellency. Repellents can act in different dimensions of effectiveness. And DEET is one of these things that works 2 ways. It works as a spatial repellent. Mosquitoes are flying, they're like, ugh, DEET. But if they land on skin that has DEET, that's the contact irritant. They're like, no thank you, and then they fly away. Some repellents only do the spatial stuff, and some only do the contact stuff. The neat part about DEET is that it does both. Contact, spatial, toxicity, no thanks. I mean, the quickest way to do this is just to jump to the methods, to the plots. Ooh, no. That's not good. There's no variation in outcomes. Okay, this is mortality. I don't need to look at this. Okay, what do we got here? We got contact irritancy. Okay, so let me minimize this a bit. Okay, so contact irritancy for the 2 species. We got the huge diversity of things tested: citronella, basil, catnip, and then the two positive controls, DEET and picaridin. Standard errors. All right, so I need to pull these data from the graph again. So I need to copy and paste this. Of course, should have been smart and set this up beforehand. I need to start R. Oh boy. I'm just going to have one graph at a time. Resize it a bit. Might be too big. Okay, so first one for 80s Aegypti. All right, let's bring it up in R. Hopefully that works. Nope. My computer is being super slow again. Let's see, have a look to see what is cooking here. Let's just keep moving. Okay, so let me double check to see if you see what I'm doing. All right. Yep. So time to extract data from this figure. That should be pretty quick. Okay, which one is catnip? It's the splotchy one. See, this graph wouldn't be good for automated extraction because it has so many lines. The hatching to distinguish between bars. Okay, good job still plotting the error on that one. All right, I got all the catnips. I'm just going to change groups here and do the DEET, which is this birch-like hatching. Yeah, that type of hatching would totally mess up. Once I'm done this, I'll press the automated button to just show you how it confuses. Because all those little hatches, you know, it's trying to figure out the error bars, and all the little hatches within the columns are like little error bar markers. Okay, last one, the picaridin. No need to be pixel perfect on this stuff. Last one for the win. Hopefully I won't have too many. Let's see these extractions. Whoops, did not include the scale. Just 0 to 100. There we go, we got our numbers. Do they make sense? Dose is at 3, yep. So I'm just gonna copy that and then put it in Excel. I'm just gonna put it somewhere. Oof, there's so much stuff going on here. We got the mean, error, access. This stuff can be deleted. I gotta make sure I have the right groups. The first group was the catnip. Second group was peat. And then the third group was the picaridin. Okay. Mean response. And then these were all done at different dosages from 0.5, 1, 2, 3. So dose 0.1, 0.5, 1, 2, 3. And then that gets repeated. Okay, so I got the data semi-organized here. Let me just double check to see if it was standard deviations. Standard errors. Oof. Okay. Well, there you go. Okay, so now I gotta start really putting this in a table here. So repellent concentration, it varied. Right, so right now I'm just doing the 80s. I'm gonna shove this in another spot because I'm focusing on 80s with all the concentrations done on 80s. I mean, this technically what I should be doing is alright, I need to insert a bunch of rows. Maybe I'm just going to be doing one today because this is already taking too long. Alright, so 0.5123% dose. How is it presented in the paper? I don't remember. Oh, I'm hungry. Mosquitoes. Contact accuracy. Consists of one clear cylinder attached to two metal cylinders containing either control or treatment. Let's see if there's a picture of what they did. No picture. So like they had it in cylinders. SRA configuration. I gotta look what SRA is. spatial, oh yeah, it's written right there. Spatial repellency assay consists of one clear cylinder attached to two metal cylinders containing either control or treatment netting. 20 females were introduced into the clear cylinder. So this is kind of like a wide tube situation, and allowed to rest for 30 seconds, after which the butterfly valves were opened for 10 minutes. Following the exposure period, the number of mosquitoes in each metal clear chamber was recorded. Okay, so this is kind of like a wide tube experiment. You got your repellent, you got your control, you see where the mosquitoes fly in. No indication of what is the attractant. Maybe I missed that. Essential oils. Looks like there's no attractant. It's just, you know, your mosquito has a choice to go into one box or the other. Experiment type is infused netting. I'll call it a Y cage. Infused netting in Y cage. Because Y, it has the shape of a Y. Two choice bioassay. Experimental temperature. Lots of discussion of how the mosquitoes were maintained. Both maintained in the entomological lab at 25, right? But no information on how the actual experiment, what was the ambient temperature in the experiment. I mean, that's super quick stuff to just eyeball because it's in Celsius, not reported. Okay, let's keep moving. I think it was 20 again. 20 mosquitoes were thrown in for the contact. We got, it's not clear. Yeah, okay, so we got positive. I did not write positive right in the other ones. Just correct that quickly. Positive. We got DEET. I guess DEET at different concentrations again were used. Yeah, they must have also manipulated the concentration of DEET, ranging from 0.5 to 3%. Okay. All right, going by quickly. Okay, what is the response type? It's a spatial repellency. How do they call it? Escape. Right, so the mosquitoes are like, yikes, repellent, and then so they fly in another box. So spatial repellency. I mean, okay. Response units. Percent escape. percentage. How long did they run the experiment for? I think it was, I wish I had a good memory, you know? After introduction, mosquitoes were allowed to rest for 30 seconds. Then the butterfly valve was opened for 10 minutes, after which the valve... Okay, 10 minutes. All right, response mean. I got all that in the table for mint. Right, I'm going to put SE here because I'm going to need to figure out to back calculate to get the standard deviations. Again, I wouldn't normally do it this way. I would have a separate column that just says SE, SD, whatever they come up, whatever confidence intervals, and then later I would do it when I'm just trying to do this fast. Okay, this is where it gets a little weird on how many times they actually ran the experiment. Contact irritancy cylinders were connected. 10 females mosquitoes were released. Okay, I got that number wrong already. The number of mosquitoes that escaped, both chambers, 6. 6, another magical number. And since I'm doing DEET as the first repellent, all this stuff gets repeated. That response, and then finally last to plug in the values I extracted from the plot for the DEET group. There we go. So now I just need to repeat this thing again for picaridin, and then repeat it again for the other mosquito, Anopheles mosquito. Alright, so this is going to be super boring. I'm just going to follow through to complete the project. Copy and paste this whole... I'm just going to... this part here gets repeated. Remember, my goal is to have one effect per row. And so this stuff here gets repeated because it's catnip first contrasted with DEET, then catnip again contrasted with the other positive control. All this stuff stays the same. But it just becomes picaridin. And I think all this, yep, all that just stays the same. 1, 2, 3, 4. There we go. Did it for the 2... Spawn, double check. Okay, now I got to repeat the whole thing again with the other species of mosquito, Anopheles. And then I got to do it all again to get the spatial repellency. So right now I'm just doing the contact repellency part. Okay, let's see how fast I can do it this time. Now I'm doing it for the Anopheles. Let's come back to R. All right, let's extract these data all over again. Double check, catnip, splotchy, 1. No error bars on this one. That's a real bummer. Deet is birch. So good again. This is because they decided to represent the outcome of the experiment using percentages. You get bounded values. There's no variation around the boundary, right? So if things are 100% all the time, there's no variation. And then finally, picaridin. Last one. Does that make sense? The values. Yep, we got a bunch of hundreds. All right. Let's move that stuff onto Excel and just finish processing this paper. I mean, everything is pretty much the same this time. but the species of mosquito is different. All right, so I'm going to copy the whole thing from 80s. Going to drop it. I'm just going to slightly change the color on it so I don't confuse it with the other one. Change the species name, Anopheles. And then just drag and drop these means. Four values for catnip. Which gets repeated twice because we have two positive controls. Let's plug in the values for DEET. Oops. And then finally for the Pickardin. And then I'm done. I think oh no, there's still the spatial repellency. I'm going to have to repeat this whole thing again for the spatial repellency. I think you guys get the picture. I'm not going to torment you guys anymore with this stuff. So let's quickly do the math here on the number of effects extracted for this study. So far we got 16 effects, 2 types of positive controls, 2 species by mosquitoes, by 4 doses. So 4 times 2, 8, 8 times 2, 16. That makes sense. You need to verify, double check in your head, and then I need to repeat again all this with the spatial repellency stuff. Go back, extract the data. I'll do it later, and then I'll process another study some other time. Okay, so one study, 3/4 processed in one lecture. I'm doing awesome. Only 22 studies to go. This is the greatest decision I've ever made, to record myself do all this silly stuff. All right, have a good weekend.